continue to watch these videos. We are uploading them on drbeen.com, and we would also upload them on BitChute as well. So just some information. Let's start our discussion. The discussion today is very exciting. Today we are going to talk about a new drug for COVID-19 called Leronlimab. This drug has been used or has been built for HIV patients, and now this drug has been offered to COVID-19 patients as well. The good news about this drug is that this can be used in all phases of the disease: mild, moderate, severe, critical phases. Look at it once more. So we're talking about Leronlimab. This is a humanized immunoglobulin 4 antibody. It is a monoclonal antibody which is humanized. And what does that mean? What it means is, look, here is this antibody. We have talked about antibodies many, many times in our discussions. So this is a heavy chain. This is another heavy chain. These are proteins. And then this is one light chain, and this is another light chain. So immunoglobulins are made up of 2 heavy chains and 2 light chains that in turn have many smaller parts. What is more interesting for us is to note this part and this part. These are called antigen binding parts or binding sites. And these antigen binding sites have special protein structures which can then bind to antigens on a virus or a bacteria or a fungus or any other harmful thing. So these sites, keep an eye on this because this is important for today's discussion. These sites are called binding regions. And within the binding region, the part that actually binds, that actually does the function of binding, the remaining part is just like a building. And this is the door in which you catch the pathogen. This part is called complementarity determining regions. This is what determines what does an antibody do? Who will this antibody attack? That would be defined by this part, and this part, and this part, and this part. Good? So far, so good? Wow, Joshua. So Joshua is saying that $2,200 for 2 doses. Now, the study that I see from them, in that study, they had given 1 dose only, and that created the results. So if that is the case, let's say $2,200 for 2 doses, that means $1,100 for 1 dose, and that might still be doable. So, Lying Eyes is asking this question that, thank you for covering it. When you learn about a drug, how do you know how it will do what they say? So that is the study, correct? So the point of the study is to bring the data to, let's say, FDA and tell FDA that, hey, look, here is our data. This is what we had done. This is the results. And FDA's researchers and doctors will sit down, look at that data, do a rigorous, thorough study of it, and then come back and say, yeah, it looks like you have something. And then they would ask them to do more of the studies. So you go from phase 0 to 1 to 2 to 3, and then finally phase 4 is actually production in or selling to us, other people. So Joshua, the answer, so Lying Eyes, the answer to your question is they have to prove it. Without that, it is not possible. So back here to the discussion, we're talking about antibodies. And why are we talking about antibodies? Because this drug itself, Leronlimab, is an antibody. It is a monoclonal antibody. And we'll talk about that in a second. Monoclonal means it is one kind of antibody, and it is produced by mostly through the bacteria and generated in mice, and then as the recombinant technology is used for bacteria. We've talked about that in the past. So is this good? So we call it IgG4 instead of saying IgG. And why do we call it IgG4? This monoclonal antibody has a specific mutation produced in its hinge region, that is this region, and due to that it is called an IgG4. And various kind of changes in antibodies can be named differently. So anyways, this is an IgG4 and it is humanized. Good. So Alexander says, Dr. Alexander, thank you very much for joining. You are a genius mind. And you're saying it is correct to say that IgG 1, 2, 3 and very different from IgG4. Absolutely correct. And then let's continue our discussion. First part of the discussion. And again, guys, please realize that when I discuss these things, I can simply say this, that Leronlimab has shown an effect in saving critically ill patients. The point of doing these discussions are to do a deeper dive into the actual mechanics of something and to be able to understand it so that you can then on your own assess various drugs, various literature, and figure out what is propaganda, what is wrong, what is right, and what is medical concept. This is why I go into these deeper discussions. So here, what is a humanized antibody? So let's look at that. What we do is, we take the antigen, in this case, let's say coronavirus. We take the coronavirus and we inject that into an animal, for example, a mouse. Now, when the mouse will produce antibodies against it, those proteins that mouse will produce in the antibody will be mouse proteins. The point is, to our body, they would look foreign. So if you take those antibodies from the mouse and then you put that in a human being, human immune system will react to these antibodies saying that, hey, these are foreign material. Even when, love the mouse hiding in the cheese, correct? So even when the mouse antibodies are going to help us, but we still cannot work with them because our body thinks that that is just like a virus and a bacteria, these antibodies are also something foreign. So we try to do what? We try to make these antibodies as nearly matching a human antibody as possible. And that process is called humanization. And what does that mean? So here we have a mouse, and this mouse has antigens produced, let's say, against coronavirus spike protein. And this is that antibody produced. I said antigen, antibody. Then here is a human being. I thought that if mouse is going to have cheese, then human being should have also something equivalent funny. So the human has his pizza with him. And let's say we have some antibody, not necessarily against coronavirus, some antibody produced by this human, and we are producing antibodies all the time. And we take that antibody. Now what we do is we kind of stitch them. How do we stitch them? We develop a DNA that has parts of the genes from our antibodies and parts of the genes from the mouse DNA that is making that antibody. So we kind of take both DNAs and start mixing them to do what? To create another antibody, which is a mixture of both. So we are at a microscopic level, we are transplanting the pieces of antibodies to make the mouse antibody look less like a mouse antibody by manipulating the DNA that generates the antibodies. Now, please remember, many people afterwards comment and say, I don't want anything that would change my RNA or DNA. This DNA is not changed in us. They make the DNA in the lab, then they give this DNA to a bacteria. Bacteria will use that DNA to make antibodies, and then we get the antibodies. So it's not a change of DNA in us. Now, when we have these two antibodies, then we create combinations of them. What are those combinations? Look, this one is half. If you see here, this part is human part and this part is mouse part. This kind of an antibody is called chimeric antibody. And normally in the drug name, there is a pronunciation of XI in it. So that is how you know that this is a chimeric antibody. Then look here, in this one, we have even more human part. Here we even have more human parts and even lesser mouse parts. So this is called chimeric because there are mouse parts in there, but humanized. So now it is becoming humanized. And these kind of antibody drugs are usually, they have CZU in their name. For example, abceximab. So they have CZU in their name. And then finally, this is the best form where only the antigen binding parts are from the mouse, these red parts, and the remaining whole antibody is human. And these usually have ZU in their name, like Zumab. And these are called humanized. So what is a humanized antibody? If I now go back here, we take an antibody from the mouse, which is against a specific virus. For example, in our case, COVID-19. We are interested in the mouse antibody. We are interested in these areas which are COVID-19 binding sites. Then we take a human antibody, any antibody, and we take these binding sites and transplant them here. And we do this via DNA modification, and then that DNA goes into the viruses as bacteria, which will then use that DNA to produce antibodies, and now we have antibodies. We can then inject them into our bodies. So chimeric antibodies, chimeric humanized, and then humanized. So this specific Leronlimab is humanized. So majority of this is a human antibody, and a smaller piece is from mouse. That piece is the one that is specific to bind to what is that? A specific receptor, and I'll talk about that. Now, where does it bind? Let's look at that. It binds with CCR5 receptors. So today we have a new area of immune system that we are discovering thanks to this drug. So chemokine receptor number 5. That is a receptor. It is present on majority of our cells. Our cells express that. Many cells do not express that. For example, I'll go here in a second, but let me first explain what this receptor is, how it is made. On our chromosome number 3, on the short arm, remember chromosome have a long arm and a short arm. Short arm are also called P arm, P for petite or small. So on the small arm of chromosome number 3, on the position number 21, there is a gene that helps our cells produce chemokine receptor type 5, this receptor. So this is a cell here. This is a cell here. This cell is producing chemokine receptor type 5. It is the receptor itself is a G protein coupled receptor. It's just a term. It's a technical thing that it's a protein. G proteins are usually 7 loop proteins that are present in the cell membrane. So the point of this all is that this is a receptor that is present on the cell surface. Good. It is called CCR5. We are very much interested in this cell, in this receptor. When this receptor is stimulated, when something comes in and stimulates this receptor here, that causes calcium channels to open up in the cell. It causes calcium channels to open up in the cell. These are the calcium channels. When calcium channels open up, calcium goes in the cell, and many of the cellular functions are activated by the calcium going in the cell. So here, for example, when CCR5 receptor is activated and calcium is gone into the cell, that results in cell activation and especially chemotaxis. Keep this in mind. So in the whole discussion, please remember the word chemotaxis, because the whole discussion is about the chemotaxis. Chemotaxis mean movement of the cells, usually immune cells, towards some chemical mediator, chemical substance. What does that mean? So imagine if I have a lot of, I have some bird food. And so here is a bird. Let's see if I can make a bird here. So let's say here is a bird. This is the beak of the bird. And this is the bird, right? So the bird, we want the bird to come here. So what we do is we put some bird food here, then a little more here, and then a lot more here. So what will happen? The bird is going to eat a few of these, you know, things, seeds, and then move forward, and then eat some more here, and then move forward. So as the concentration of the bird seed is increasing, the bird keeps moving in that direction. Finally, it reaches the goal where we want it to go. This is the process of chemotaxis. If instead of a bird, if we have a, let's say, macrophage here, so this is a macrophage, and you have chemical substances that macrophage is attracted to, and more concentration is where there is a fight going on, inflammation, infection happening, then lighter concentration here, then this macrophage is going to slowly move all the way to the higher concentration and come here and take part in the fight. This is so Carol said it is a duck. Yes, I thought it was a bird. Isn't duck a bird as well? So this is what is called chemotaxis. Chemo mean chemical substances, taxis mean movement. So moving with the chemical substances, reaching towards a goal. And that is what we want. Whenever our cells are breaking down, let's say we have COVID-19 and there is an inflammation going on, an infection going on in our lungs, and the cells are breaking down, they would release chemical substances. Those chemical substances are going to ask other cells in the remaining part of the body to come over there as well by chemotaxis. Now, for the immune cells to work by chemotaxis, they must have CCR5 receptor on them. CCR5 receptor, just like the children's toys. So let's say this is a remote control car of a child, and then there is a remote control. So this antenna on the car can be CCR5, and the remote control can be CCL, chemokine ligand, and when or the signal can be CCL. So when the signal is going and it hits the antenna, that car is going to start moving. That is the same thing here. A cell, an immune cell that can move, needs to have a CCR5 receptor on it. And when a chemical substance would connect there, then the cell would start moving by getting calcium in its body, in the cell itself, and that calcium is going to make the cell walk. So this is the process of chemotaxis. Now for the CCR5 receptor, the ligands are the things that can attach to that receptor and make it work. Ligands are called CCL3, chemokine ligand number 3, ligand number 4, and ligand number 5. Here in this study for Leronlimab, these two things are important, CCR5 and CCL5. So there is a very good question in the chat here. Lying Guy says that I thought that the virus shuts down the signal for help. No, actually, Lying Guys, the virus accentuates the signals for help, causing so many of the inflammatory cells to come over, causing so much of the fight to happen, unnecessarily more, disproportionately more, that causes our tissues to be damaged. Alexander says, calcium, vitamin D, how important is vitamin D? Very important. So without vitamin D, all of this function does not happen. So without vitamin D, immune system is not going to help us. So Dr. Alexandros, you are very correct. So back here, ligands are antigens? No, ligands are actually produced in our body. So let's say, let me make it this way. Imagine here's a macrophage. We want this macrophage to come here where there is a cell which has been infected by coronavirus. Right? So this is a cell, normal host cell, let's say lung epithelium cell. It has become infected by coronavirus. It is crying now. And in this process, what the cell is going to do is it's going to release CCLs, CCL3 or CCL4 or CCL5. Normally CCL5. CCL3 and 4 are only released by a smaller number of cells, but CCL5 can be released by almost every cell or a majority of the cells. So the cell, when it has, let's say it is our lung epithelium cell, it got damaged by the coronavirus. As a result of that, the epithelial cell released CCL5. That CCL5 is going to act like a bird food for the macrophage. Now, macrophage is going to have a receptor on it which is CCR5. CCR5. So R for receptor, and this guy is CCL5, as L for ligand. Ligand is something that can attach to that receptor. So when this ligand goes here and attaches to this receptor, that makes this macrophage move towards the ligand. This is like the remote control, but it's a chemical signal instead of a electromagnetic signal. So to answer your question, that is the CCL an antigen? No, it is not an antigen. It is a substance, chemical substance produced by our own body. So far so good. So now back here. Beautiful mechanism that we are discussing. So CCR5 is present. CCR5 is an important ligand for that. The CCL5 is now released. If we come here, first of all, let's see where the CCR5 are present. Where are the remote control antennas? They are present on T cells. So if there is CCL5, then T cells are going to move. So there is a question, Holy Angel, Dr. Bean, so the CCL5, etc. are cellular debris. They could be debris, but they are actually intentional releases by the cell as well. Some cells, when they are stressed out, they release CCL5 without getting dead, without becoming a debris yet. So now the CCR5 receptor is present. So look at this little bow. Are present on T cells, they are present on macrophages, and they are present on dendritic cells. Dendritic cells are also immune system cells. Now, important thing here, CCR5 are usually not present in the breast epithelium. And epithelium will mean what? The part, the canaliculi of the breast that bring milk out, they are exposed to the external surface. So all of that canalicular tube which makes the milk and brings it out is lined by epithelium. Breast epithelium usually does not have CCR5 receptors on it. However, in women, 50% of the breast cancers, these cells develop CCR5 on them. And when they develop CCR5 on them, what happens is that these cells, remember the CCR5 receptor is usually present on the remote control cars. And when you activate them, the car moves. Same way when CCR5 is present on a cell, if you activate that receptor, the cell is going to start moving. This is how the metastasis occur. This is how the cancer cell move from, this is one of the mechanisms, there are many. This is how the cancer cells will move from the breast, so breast cancer cell, and go somewhere else and cause cancer. So this drug can actually bind to these CCR5 receptors and block them. And when it blocks them, then the breast cancer cells would stop moving. They would stay in place. And now what is that? The other drugs that are cancer killing drugs, they would come and kill them because these cells are now sitting ducks, right? So chemotherapy would come and kill them. So this is another area. And 50% of the triple negative breast cancers are actually CCR5 plus. So this is a very important thing. Similarly, prostate cancer. Prostate cancer, in the cancer cells also have CCR5 receptors on them, and that also allows them to move. So it looks like that CCR5 receptor, whichever cell has them, is usually the cell that would move. And we do not want the cancer cells to move because they will go to other places and they would cause further cancers or spreading of the cancer. And they have found that if they give Leronlimab and they make these cells not to move by whatever mechanism, this drug or some other way, then they can give chemotherapy and kill the cancer cells and get out of the problem. Then, one more thing, this drug was actually made for HIV. And how did it work? So this is the HIV, this is not coronavirus, this is a retrovirus for HIV. This virus binds with the CCR5 receptor. And when it binds there, that is how it enters, just like COVID-19 binds with ACE2, right? So here, the HIV virus binds with the CCR5 receptors. So this company had made this drug, which is an antibody, and it would come and bind. Remember, antibodies are like Y-shaped. So it would come and bind with the CCR5 receptor, and it will block it. When it will block that, then that cell cannot be, that ACE, I'm saying CCR5 receptors cannot be used by the virus to get into our body. Just like nowadays we are contemplating that if we have angiotensin receptor blocker, and that receptor occupies the ACE2 receptor enzyme, then the coronavirus cannot connect and go into the cell. That was the same mechanism here for this drug. So the point of this is that CCR5 are present in many places, and then the drug was built basically for HIV. Now, let's see, how did this drug actually help out for how does it help out for the COVID-19? So let's look at the study here. So couple of things. This is the research, this is the study that Leronlimab company, they did this study. So this is that. I'm going to go over this. But in addition, I wanted to show you that there are 2 trials now that have been approved. One is here, study to evaluate the efficacy and safety of Leronlimab for patients with severe or critical COVID. And this one is study to evaluate this for mild to moderate. And this is the good part about this drug, that it can go from mild to all the way to the critical and can be applicable in all phases. So hydroxychloroquine, for example, asymptomatic or mild or prophylaxis. Similarly, remdesivir, for example, could be in the early phases, but mostly in the later phases. This drug can actually cover a majority of the phases. Then here is a discussion of what is CCR5. I would put those links out back here. Here is the study that they did. What they said was, emerging results indicate a dysregulated immune response characterized by runaway inflammation, including cytokine release syndrome or cytokine storm, as a major driver of pathology in COVID-19. Now, what they did was they took 10 patients which were terminally ill. This is an important thing to note. Most of them were on mechanical ventilation or on oxygen therapy. So they were terminally ill. And these patients were given these drugs. And I think it was given once. 4 of the patients died, but the usual death rate here in the New York was 88% of the ventilated patients. So instead of 88% dying, 40% died. And even for those, they have an explanation for what happened. But the remaining 60% received the treatment and became okay. Now the question is, what was the treatment? So let's look at what they have been saying here. So what they are saying is, look, CCL5, forget this 8 and 9 here. This 8 and 9 is the reference number to the study, but that is just not a superscript. So it is CCL5. So they talk about CCL5 and CCR5 axis. And my apologies for the cat. I'm going to close my door and see if that helps. Okay, back here. I don't know if Luffy would stop chatting, but please forgive me. Here what they did was, they had administered the Leronlimab once to 10 patients. That is what they had done. Mark, I'm so sorry. This is the fun of working from home. And what happened was, so here they're saying that the mortality rate in New York is 88%. In the case of the critically ill, terminally ill patients that received Leronlimab, 60% were saved. So that is a big difference. Every life saved is a big difference. And here is what their basic mechanism of action is. So I would let you read these, but look at the dose here. They have given subcutaneous 700 milligram injection of Leronlimab. And I'm going to explain what happened. The basic idea is this. When the coronavirus enters our body, our lung epithelium, that is the trachea, the epithelium here, and our macrophages that are fighting there, that were already present there. They did not come over there. We have macrophages and immune cells present in almost all tissues, including lung epithelium as well. So the resident macrophages and the resident T cell over there, they will all start making this reddish thing that I made dots here. These dots are CCL5. So these are the signals to the immune system to say, we are under stress, we are under destruction, please come and help us. So this is CCL5. And this CCL5, as I mentioned before, is now going to act as a chemotactic substance. And how would that happen? Here is the army of the immune cells. What are these cells? There are T cells here, there are B cells here, there are neutrophils here, there are other macrophages, there are thousands of millions of cells present in our lymph nodes and in our bone marrows and in our body tissues. They all have the CCR5 on them, receptor for CCL5, the chemical substance. So they're all standing there like remote controlled cars and saying, okay, give me a signal and I'll come over to fight. And here the signal is now being generated by the immune cells or by the cells that are under attack. May these be epithelial cells or immune cells. When that signal finally reaches these guys, the immune cells, and stimulate their CCR5s, they would all run towards the area of the fight. And that is how this whole immune system cells, they would all come here and start fighting. The result of that, and we have talked about this at why, so there is a video I've done, why COVID-19 kills so fast. The result of that is that there is a lots of release of interleukin-1 and 6 and 8 and tumor necrosis factors, and I do not know what else. There are so many chemical substances released which cause further amplification of immune system, which cause severe destruction of the lungs, and we go from pneumonia to ARDS to septic shock to death. So this whole process, the axis in it, the basic backbone of that is CCL5 and CCR5. CCL is the chemical substance or the ligand that would go to the immune cells and tell them to work and come over. CCR is the receptor present on the immune cell. When you activate it with CCL5, the immune cell is going to walk like a remote controlled car to the area of the fight. So imagine if we don't have CCR5 function, if we block these receptors, and do you think that these immune cells would run towards the fight? They will not. Why? Because they're blocked. So CCL5, their activating ligand, cannot come and bind there. That is exactly what Leronlimab is. So now how did they try that? So they took patients, and in those patients, pay attention here please, that they had increased CCL5. That is a ligand, that is a chemical substance saying, hey guys, come over, help me. So their CCL5 was 100 folds more than regular people. So there was a huge noise going on in their body to say, please bring all the immune system to the lungs and let's have a fight. Their interleukin-6 was increased, which is terrible in the inflammation. Again, it is great that it helps us take care of the infection, but when it is produced in more amounts than normal, then it would cause even more chemotaxis of the immune system. It would cause even more production of the acute reactive proteins by liver, and that would cause even more damage. So interleukin-6 was increased, CCL5 was increased, leukocytosis was less. So leukocytes were less. So leukocytopenia was there. What does that mean? The excuse me, the white blood cells were being destroyed in the area of fight. The soldiers were being killed by the virus. So leukocytes, the white blood cells, our army was being killed. So they were less in number. So leukocytopenia was occurring. Renal damage was there, liver damage was there. So remember, they gave drug to those patients that were having visceral damages. So these were not the patients who were on the right track. They were going downhill. They were dying. And then what they did was they injected 700 micromilligram of the drug, and result within, I love it. Look at the results. Within 2 weeks, 6 patients were recovered. 4 patients had died. Within 3 days, interleukin 6 level went down. That means it calmed down the inflammation, the cytokine storm within 3 days. Then the interleukin 6 levels reached the healthy levels within 14 days. Beautiful, beautiful. Then, as much as it is an expensive drug, as much as this may be big pharma, I do not even know they're big or small pharma, at least the mechanism is great. So please don't start thinking that I'm a big pharma shill and start commenting there. It's a great mechanism to know. Maybe if you can understand this mechanism, maybe you make this drug. Maybe you are a small pharma and you make it. But at least know what the mechanism is. So here, the other thing that it did, the drug, it made the CD4 to CD8 ratio correct. So that what that means is it calmed down the immune system so much that the normal cellular immune cell ratios and numbers started regenerating, which is also very important. CD8 levels were corrected as well. And CD8 are necessary to kill the virally infected cells. So this was a great outcome. And the result of that was, so let me just stop here for 1 second. Do you see that what a beautiful mechanism it is? That they disrupted the signaling from the damaged cell to immune cells to come over to the site and fight. They disrupted the extra unnecessary disproportionate action of the immune system, which ends up killing us. So, Holy Angel, the mechanism of the drug, I'm sorry that I did not make it clear. The mechanism of the drug is that, where was it? So here, this drug, I had mentioned here that this is the CCR5 receptor, correct? This drug blocks this receptor. That is what I had discussed somewhere over here. So Leronlimab blocks the CCR5 receptor. Because it blocks that receptor, the cells cannot respond to CCL5, which is the ligand, and asking the cell to come over. So now the cells are not going to respond to CCL5, and that is how they would not move. So Leronlimab, if it was a Y-like shape of the drug, it would now block all of these CCR5s. When it blocks them, then the CCL5 cannot connect there. And when CCL5 cannot connect, it cannot move the immune cells. Isn't that beautiful? So are destroyed leukocytes a source of CCL5? Yes. Most of the cells can produce CCL5, either living or destroyed. Most of the living cells produce CCL5s. What are the complications that can occur from this? So that is a very good question. Jennifer is asking that. So let me show you this. They said that we have used this drug in HIV patients, where 800 HIV patients here. HIV clinical trials where over 800 individuals have received Leronlimab, no drug-related deaths, serious injection site reactions, all drug-drug interactions were reported. And here is the deal. The CCR5 is not a very important receptor to be present everywhere and to be active everywhere. It is important for the immune system to function, but remember, it appears in the cancer cells as well. And in case of COVID-19, there is an extra activation of that. So if we can block that, it does not hurt us too much. It only calms down our immune system. Of course, therapeutic dose has to be there, which means a dose that does not block all of them forever, but only blocks an amount that is necessary to prevent the disease, but also lets the normal functions of the body to continue. So that is what is the very good question. Jennifer had asked that what are the complications. So Holy Angel says that how does a drug actually block this? So let's talk about that as well. I have some great questions today. So look, when we make an antibody, so let's say this is an antibody, I'm making it now the other way around. Antibody has the binding site. That binding site is usually specific to an antigen. So let's say we take our receptor. This is our CCR5 receptor, CCR5. And there is some pattern of proteins on this receptor. And we make the Leronlimab, we make that protein, the antibody, in a way that it can bind to this pattern on the receptor. Then what it does is it simply comes and mechanically connects with it. And that covers the receptor, makes it unavailable for the ligand CCL5, for example, who was eyeing this area to connect. That cannot connect here anymore because it is occupied by Leronlimab. That is how it stops the action. Sunil is asking any contraindication. I do not know of any contraindication so far because the function is to calm down the immune system, but very selectively calm down. It's not an immunosuppressant, but it is an immunomodulator, and it disrupts the cytokine storm by disrupting the CCL5 and CCR5 interaction. So they call that as an axis. What does that mean? If we go back here, and thank you very much for such great questions. If we go back here, first look at it this way. Damage occurred. Damage caused the CCL5 to ask the remaining immune cells to come over. Then the immune cells that come over, so now they're all over here, more immune cells, they're going to create even more CCL5s, which would go and bring even more immune cells over here, which would now have even more immune cells here, which would produce even more CCL5. And this is the immune system overdrive or the cytokine storm. And they break this cycle. They disrupt, according to them, they disrupt the cycle. So I love that. What a beautiful attack. Somebody thought of this, that, hey, why not we do this? And then they did it. This is why I keep saying that the whole world has become equal because of this COVID-19. And some people said that, no, that's not the case. But look at this. There are some developing nations that are actually doing better than some modern nations. And the chances to think and the chances to figure out a solution is now equal for everyone. We are sitting here, all of us are not doctors. Many of the audience here, they tell me that they are not doctors, but they are thinking, they are learning, and they are able to offer insights for what can be done. So that is what is education and science. So I love it. So continuing on, I'm impressed by the question, so I am talking a little more about that. So this is what that drug did, and 60% of the patients they survived. And not only that, they recovered anywhere from 3 days to within 14 days. Normally, people who are on ventilators, I have a friend whose brother is here in Coney Island, unfortunately on ventilator. He has been on ventilator for 34 days. He opened his eyes yesterday after 34 days. And here, these are the patients that were undergoing liver damage and kidney damage and had cytokine storm going on, and they were on ventilators, and they gave the drug and they started getting up and going home within 3 days to 14 days. That is beautiful. So continuing on, what else does this drug do? So one is that it disrupts CCL5 and CCR5 axis and stops the cytokine storm. Then what else it does is that in the COVID-19 infection, they found out that the bone marrow, so this is a bone, this is a vertebra. Bone marrow of this vertebra was making the cells in the bone marrow. That means these are the cells that are going to make more white blood cells and neutrophil and macrophages. So the new army that was being produced in our bones, they found out that that army had cells that had interferon genes open in them. That means they were getting ready for tremendous fight. They had CCL4, CCL3, CXCL8 gene open in them. That is another tremendous fight that, hey, we're going to signal so much with the ligands that we're going to bring the whole immune system over there. So the new cells that were being formed, they were already being formed with the enhanced signaling mechanisms in them to say, go fight and call everyone there. And they had lots of interleukin-1 and CD69. CD69 are T cell. Those genes were open. So new cells were being manufactured that would enhance the immune system even more. And what happened was that when they gave Leronlimab, they saw that even these genes that were open, that genetic mechanism was suppressed as well or disrupted as well. And here, the CCL3 are released by T cell. CCL4 are released by T cell. So when they saw that there are more genes that are open for CCL3 and 4, that means more T cells were being formed. When they saw that there are more genes that are open for 69, that means more T cells were being formed. When they saw that there are genes that are open for IL-1, that means more macrophages were being formed. When they saw that interferon are open, that means more macrophages were being formed. So they saw in the bone marrow more fighters were being formed, which would have created even bigger problem. So once again, it was necessary for CCR5, the Leronlimab, to come and work. CCL4 is also released by neutrophils, monocyte, B cell, T cell, fibroblasts, endothelial cells, and that. So many cells can produce these ligands. CCL5 in the same way. Then what other mechanism did this drug do? What it did was, look, here is CCL5. CCL5 chemokine ligand. CCL5 can activate a T cell, an eosinophil, basophil, leukocyte, platelet activation, natural killer cell proliferation, natural killer cell activation. Almost the whole immune system can be amplified by the function of the CCL5. So, of course, remember that this Leronlimab is going to act on the receptors on which CCL5 acts, and that is how CCL5's action will be disrupted, and people start getting out of their weird or critical state. I feel so happy about this. So one more thing that it does is that it reduces the platelet activation. And how does it reduce that? Remember that calcium channels are opened up when the CCR5 is active. So when you have blocked the CCR5, the reaction in the platelets is that their activity needs calcium and they reduce the activity. So remember the hypercoagulability state? The hypercoagulability state is also reduced. That is an extra non-immune benefit of this thing, which is very useful for COVID-19. So platelet activation is reduced. Then finally, one more thing, and that is they observed that within 7 to 14 days, viremia, that is the virus present in the blood plasma, had reduced. And what does that mean? That means that the drug had worked so much on balancing out the immune system that immune system not only got balanced, but it started taking care of the virus correctly. And the amount of virus, the load of virus in our system started going down. And of course, the people got up. I mean, this is beautiful that a patient who's comatose, who is on... Okay, so this is the discussion. I have a question here. The stigma says that CCR5, are you talking about Leronlimab? Yes. So what is your question? I didn't catch your question. So Leronlimab eventually resulted into lower virus load because it balanced out the immune system. That is one. Number two, but the most important part was it disrupted the chemotaxis of the immune cells. So immune cells did not move that much. They did not arrive in the lungs that much and cause damage. That was the primary function. Second, viremia is reduced. Third, in the cells that are being formed, the body formation reduced because the immune system calmed down. The messages to bone marrow to say, bring in more cells, they reduced as well. So in so many ways, it helped the patients. And I love it. I have the link. I'll put that in the description, or it is already in the description. Please, I would encourage you to read their study, how it worked. And now they're starting the FDA has already given them approval as an emergency investigational drug to be used on compassionate basis. And they have started the trial as well. In one trial, they're looking for 370 or 50 people. In the other trial, they're looking for 75 patients, and they're going to work with them. I am sure that they would save their lives. It is an expensive drug. That is my only gripe here. So this is the discussion. What about if patients had heart, diabetes? And so all of these patients, Chanchal, they had some sort of comorbidities. That is why they were on the ventilators, because they had, so look, they had comorbidities, all of them. They were on the ventilators. They were having liver and kidney failures. So they were in a critical state. They were going to die, and they came back out of that. So there's a question, should we worry about blood bank contamination with coronavirus? That is a good question. What I read when I did the blood groups and when I did the convalescent plasma, I read that the protocols that they have, most of them are able to take care of the viruses, that the viruses are not moved or not present in the blood. However, in the fresh frozen plasma precipitate, which is the really smaller part of the plasma that for the hemolytic factor, for the blood clotting factors, that process of blood collection and processing cannot take care of the viruses. So over there, there is a problem. So normally I would say that if it is at least in the countries where the blood collection processes are correctly adhered to, they should be okay. Okay, so there is a question, will it get FDA approval? Yes, it has already gotten the FDA approval for compassionate use as an emergency investigational drug. Then Dr. Yo is Louise Fran has been fighting for Leronlimab. Who is Dr. Yo? Someone? Leronlimab is great. So we looked at the mechanism itself. My way of working is that I just show you the mechanism. You can make up your own mind. After that, you say, I don't think it works, fine. If you think it can work, fine. So without it, 40% I will correct. And I think that they feel that if the drug was given early on, even that 40% may not have happened. So due to that, they're doing the trials in the mild and moderate states as well, in addition to the critical state. So again, will this get FDA approval? Approval is already given. It is about $1,200 a treatment. In my opinion, not too expensive. I agree with you, Peter. $1,200, one dose for saving a person's life on a critical stage, where I think every day's expense for that patient is more than $1,200. So you are correct. Daniel, thank you. Can we give all the drugs on daily basis, or we have to check the stage of virus? No, so this drug can be given mild, moderate, severe, critical at any time. How expensive? So people are saying that $1,200 or so, which is not very expensive to save the life. So this drug looks like have a role in the control of RANTES? Absolutely. So the CCL5 work is all RANTES related. So you're correct. Can it be used in non-critical cases? Yes. So they are doing a study on mild to moderate as well. Dr. Bean talking about, you're very welcome. Thank you. You're very welcome. Is there a difference between Asia coronavirus and Europe? No, it's the same. There is a difference in response. Asian countries seem to have lower spread and lower death rate compared to the European countries, but that is not the coronavirus difference. That probably is the temperature and the climate and the hygiene theory and other things. Maybe BCG, which I do not know. BCG is the reason or not, but there are reasons. I am looking for Asian countries doctors to come up with the studies to say this is why there is a difference. Leronlimab seems cheap compared to the other potential treatments I've read about. Absolutely, absolutely. And it is, I think, just one dose. Do you think this will get more attention? I think so. Worth it, absolutely. I think so. What is the rate between IL-6 and RANTES? Let's do that at a different time. We're almost 1 hour into it. But don't you like this, that when I share this with us, I go deeper into the discussion of it so you can actually make up your own mind and think about it. I actually like that, to just share the whole concept. What are the side effects? Not known yet. And as I showed you the study, they said that in 800 patients of AIDS, when they gave this drug, there were no side effects, there were no drug-to-drug reactions, and there were no health issues. If the trials are successful for Leronlimab, how soon can we expect FDA, EUA like this? So they've done it. They have already given go-ahead for compassionate use and go-ahead for phase 2 trials. So it is available. What about IL-6 and CCL5 in bloodstream? No damage or nothing that will increase release when there is no immune risk. So IL-6 was increased because there is immune response, a disproportionate immune response. So IL-6 went down because the immune system calmed down. CCL5 did not go down because the immune system cells were still making CCL5, but because the drug had blocked the CCR5, receptors were blocked, even if the CCL5 was increased, it didn't do anything. So no issues. Thank you very much. Love you like this. Thank you very much. Great. Thank you very much. Would you please define cytokine storm? So I have done a lecture. Please watch my video. It is titled Why COVID-19 Kills So Fast. In there, it is about 30-40 minutes video. I have discussed how cytokine storm works. It is very difficult to explain that within 1 minute. I would simply say disproportionate response of our body, so aggressive that instead of just killing the pathogen, it kills us too. Love your discussion. Thank you very much. You like it? Great. Would you please define the cytokine storm? What about, so I think we have answered most of these. I love these discussions. I get a lifetime membership. Thank you very much. Although I have never tried to sell here, because I think it is necessary for all of us, laypersons, medical students, nursing students, doctors, for all of us to learn together to protect ourselves and our loved ones, and to understand what societies are doing in general, and to be able to separate out propaganda from actual information, from something that may be harmful information to useful information. That's why I do it. That's why I especially try not to sell at this time as well. But thank you very much. Whoever is